that in the near future. So next up, we're going to jump to our second partner, Lonza. We will hear about what Lonza does, and especially what Lonza does with the startups in the program, from Senthil Ramaswamy, the Director of Cell and Gene Technologies of the R&D department. So Senthil, it's a pleasure to have you today here, and we're looking forward to your insightful presentation.
Okay, great to be here, and please do let me know if you can see my screen. It comes up there.
Yes, now it comes up.
Okay, very good. Let's give a second here. Okay, great. So what I'd like to do is just take a quick moment to share a little bit about Lonza Pharma Biotech, and end with maybe the specific focus that we have in one aspect of the participation with the startup Creasphere. So at Lonza, our purpose is quite simple. Our goal is to work with our customers to make sure that we are able to help them with the needs of their patients. And so we keep our purpose very clear and very simple. The way we do this is by supporting the entire development cycle. So very often our customers are at either end of the development cycle. They have a new product or a new therapy or a drug that they're working on, or they are then supporting the marketing of this therapy. And we essentially cover the entire range from supporting them with the needs of their drugs or therapies at the start, all the way through the development phases and into commercialization by providing them with the development and manufacturing capabilities that they require in order to make that happen. And through this, we reach a very large number of patients that our customers have, and support the success of the therapies there. So over the last several years, and this is no surprise, the healthcare industry, specifically from a drug development perspective, has had a lot of success. And our purpose is to anticipate the needs of our customers and work with them in order to continue this great success that we've had. Very importantly, there have been very drastically improved patient outcomes in the past several years. We all, including myself, likely have friends, family, and others that we know that have benefited from the life-changing impact that new technologies in the therapeutic field have had. And Lonza is also very focused on making sure that these therapies, novel therapies, have great access to emerging markets as well. And along this line, we are constantly looking for these new technologies and being able to support our customers with the implementation of these new technologies and making them a reality. And through this, we have had the opportunity to support millions of patients by manufacturing therapies for our clients that have advanced the well-being of millions of patients. Just in 2019, I know this number is a bit dated, but just in 2019, we have over 1,000 molecules that we have supported at Lonza, and we are very proud of this, and want to make sure that we continue this journey along with our partners and our customers. And the way we want to do that is through our vision, which is delivering the medicines of tomorrow today. And we've always been in the position of predicting and adapting to a changing market, which is very relevant to what we want to do with the startup Creasphere. Just as a quick picture, we have Lonza was one of the first to provide contract manufacturing services for small molecules. And we have continued on that journey ever since through a number of different modalities. For example, we were also the first or one of the first to commercialize antibody drug conjugates, for example, in 2007. We have had significant growth in cell and gene technologies and in supporting the growth of that industry and industrialization of that industry. And we've continued focus now on more advanced therapies that are rapidly coming forth, which is one of the areas of focus for us in the Creasphere. So in addition to the technologies and really the breadth of capabilities and expertise, the way we make sure that we also achieve the goals that our customers have for their partnership with us is by our global presence. We have 29 sites across the world, both manufacturing and R&D, and we bring all of this to bear in order to achieve the mission really of making therapies and therapeutics accessible to patients that need it really anywhere in the world. We are present in 3 different continents and are growing rapidly as well. So this brings me to the area of focus for us in terms of the activities that we have in the Creasphere. One of these areas is exosomes. We see this as a rapidly growing and hopefully very successful therapeutic area. We have established capabilities for exosomes research and manufacturing in a number of our sites. Just a quick overview, I won't go through all of these. I'm actually based out of a Maryland site where we have focus on developing novel capabilities, GMP capabilities for both media and ultimately also for exosomes in a therapeutic focus. So the way we do this is to provide capabilities for exosomes manufacturing and characterization. And at Lonza, our expertise at the moment and our focus at the moment is very much on characterization, which we see as the bedrock of extracellular vesicle therapeutics. We need to have the characterization tools in order to be able to be successful in developing the right processes for our clients. And so we are looking for a partnership in the area of manufacturing platforms and technologies, which is where our work with MD Immune that will be sharing their technology next comes forth. And we have this really nice symbiosis where we provide characterization expertise, which is where we are focused to support a better understanding of their technology and hopefully translation of their technology into therapeutic outcomes. So with that, I will end my presentation here and hand off back to Fabian and the Creasphere team. Thank you for the time. Also happy to answer any questions if there might be.
Perfect. Thank you so much, Senthil. It was always a pleasure to hear your insights. And I think it is also something you said about services you provide that we as an individual sometimes don't see or forget about. So I think with that being said, it's great to have the opportunity to give those companies in that field also the spotlight today. So with that being said, I'd like to jump into the first pitch of today for MedImmune. So MedImmune has developed a drug delivery platform called Biodrone. What's the capabilities of Biodrone in particular and how they come together with what Senthil aforementioned? Seung Wook will tell you a lot about it, but first we will see a short 2 minutes video.
At MD Immune, we are developing a novel drug delivery platform called Biodrone. Using the proprietary extrusion method, we obtain tiny vesicles from cells, then we manipulate these vesicles by loading drugs or modifying surface to finally achieve targeted drug delivery. Lonza has been meeting with us on a bi-weekly basis to discuss the project goals and progress. Particularly, Lonza has provided their excellence in the single particle analysis to precisely characterize the biodrone in this 12-week program. Compared to other EVs like exosomes, we can produce far more nanoparticles in a much quicker process. Made of natural human cells, biodrones are safe and can penetrate cell membranes and tissues fairly easily. Finally, biodrone platforms can be applied to virtually any cells in any conditions, which exosomes do not currently exhibit. Hopefully you can see my video and hear me okay. All right. So hello everyone. Before I start, big thank you to Fabian and Christina and all the members at the Plug and Play, and of course our sponsor Lonza for great support. All right, here we go. So previously we presented the unique ability of our biodrone technology to produce nanovesicles in a large quantity and its potential as a novel drug delivery system. Starting our journey at the startup Creasphere, we and our sponsor Lonza met together to identify the key objectives to best assess its potential as a drug delivery system, including comparisons between biodrones and exosomes. As previously demonstrated, CDVs can be produced in far greater numbers than exosomes, probably because CDVs are derived from multiple membrane sources. We confirmed this by several means. It turns out that some membrane components are well conserved between the two, while other organelle markers, or even some classical exosome markers, are overrepresented in CDVs. For instance, some membrane proteins like CD63 are enriched in CDVs, while CD9 is scarce. This result was verified by Lonza through nanoscale flow cytometry. Similarly, we have found some unique marker sets in CDVs, which we have planned to analyze at the single particle level as well. Next, we examined these vesicles to understand how their unique properties might affect their potential as a drug carrier, first by looking at their shape. As you can see, it's nearly impossible to distinguish between these four circles. Although two of them are exosomes and the other two are CDVs, another exciting property of the CDVs is that they can be made into a powder and be quickly reconstituted by adding water while retaining their shape and biochemical properties. We also verified that vesicles maintain the original membrane topology of cells. Then we looked at individual nanovesicles to classify them with their surface markers. Initially, we used 3 classical exosome markers and found that approximately 12% of CDV particles contain those marker sets, compared to the much higher proportion typically observed from exosomes using a similar assay. This again indicates that CDVs have unique compositions compared to exosomes. We believe that the more unique marker panel for CDVs will further identify the entire subpopulations that constitute CDVs together. But whether they can be a good vehicle will ultimately be determined by their capability to load various cargoes or ability to deliver them to a specific location in our body. Over the past few months, we've been able to further improve the efficiency of RNA loading onto CDVs. Again, we confirmed that target genes are effectively suppressed when cells are treated with small RNA-loaded CDVs. When exosomes and CDVs are compared side by side, CDVs from 2 different cell sources showed similar or higher loading results than exosomes obtained from the same cells. On the other hand, by expressing membrane proteins on the surface of cells, we could obtain CDVs with a high level of target protein expression. And when cells deficient with this protein are treated with such CDVs, cells regain their lost function. Next, we added to cells CDVs or exosomes labeled with fluorescence dye. We found that recipient cells intake more CDVs than exosomes, as shown by flow cytometry or confocal imaging, while the intracellular trafficking pattern looks very similar over time between the two. We observed similar results from diverse cells. This even holds true for CDVs reconstituted from powder. We also found that RNA cargoes loaded with on the CDVs can enter these cells efficiently. In addition, when we injected CDVs into the eye, we found that CDVs can penetrate retinal tissues quite effectively and reach the epithelial barrier that has been a target for many debilitating eye diseases. Taken together, CDVs showed similar or better cellular uptake or tissue penetration results compared to the same number of exosomes. To summarize, we demonstrated that Biodrone has the potential to outperform exosomes as a drug carrier with its unique productivity that outnumbers natural exosomes. Along the way, we furthered our understanding to the finest single particle level, which will facilitate purification and quality control for large-scale manufacturing. Our next step is to prove the efficiency of our Biodrone platform at a more sophisticated level and demonstrate the functional consequences of such a Biodrones in various disease models. Finally, I would like to thank all the amazing members of MDimmune who have made this possible despite the challenges caused by the pandemic. I'd also like to thank all the project team members at Lonza for their excellent support, and of course, Plug and Play for making everything possible again. Thank you.
Thank you very much, Seung-wook. It's always amazing to see what our startups accomplish with our partners, and especially also for you in particular to see how those CDVs are well competing with exosomes and are superior in certain characteristics. So we already have the first question coming in, and that is around your development challenges, and how do you plan to what are development challenges and how do you plan to overcome these?
So certainly there are areas to improve, of course, especially to fit to the large scale manufacturing requirements, such as purification or drug loading in the industrial scale. Indeed, we are actually developing GMP compliant CDV manufacturing process already with European partners. So I think we're gonna continue to develop this global partnership to facilitate these processes.
That's fantastic. One more question, like from the audience, and it's around Biodrone as a platform, and also how Biodrone is, or what's the competitive advantage to other conventional drug delivery systems or platforms?
Sure. So I think one of the very important feature of the Biodrone is that it is made of natural human cells. That means that it should be, more biocompatible than many other drug delivery vehicles, and also Biodrones can accommodate, diverse drug modalities potentially, and then accommodate a lot of like endogenous and exogenous engineering ideas, which other DDS doesn't provide.
Very interesting. Thank you so much, Seung-wook. I know it's very late right now for you, so thank you for dialing in at such a late hour.
All right, my pleasure.
Great.
Thank you.
Okay, so for everyone, again, we're going to have all the, 1 second. Technical issues here, but that's completely fine. For everyone again in the audience, we also have the capabilities and have those startup booths, so please use them. Go to all the startups and ask any questions that we now didn't address, but will address or will be addressed during those meetings. With that being said, I would say, oh boy, we came from Berlin, traveled all the way to Chile, then traveled back to Seoul, and now we're with VelitaCell in Ireland. So I'm very happy that we have VelitaCell on board. They've developed a solution which relies on predictive analytics. And for those predictive analytics engines, they've shown that there's a never seen level of quality for cell-based biomanufacturing processes due to that platform. So with that being said, Stephanie, it's great to have you today on board. We're going to see a short video and then we can't wait to see your presentation.
We create analytical products and technologies that enable our partners to bring their life-saving medicines to market faster, cheaper, and with greater regulatory confidence. We believe that better biomanufacturing analytics are needed if we're going to meet the growing societal demand for next-generation therapeutics. With this in mind, we target multiple elements of the biomanufacturing process. Lonza are a big player in contract biomanufacturing with extensive experience in biologics, cell therapy, and exosome manufacturing. Through the Creasphere program, we've gained valuable insight into how ValidaCell technologies can support these processes. We aim to collaborate with Lonza on the application of one of our core technologies called chemstress fingerprinting to improve cell culture media design and overall biomanufacturing. The Creasphere program creates a fantastic network of established multinationals, innovative startups, supporting services, as well as members of the investment community.
Hello? Can you see my screen? Can you hear me?
Yes, we do.
Fantastic. Thank you very much. Excellent. Thank you. Good afternoon, everyone. I'd just like to start by saying thank you to both Plug and Play and Lonza for this opportunity. My name is Stephanie Davies. I'm the head of science cell therapy at ValidaCell, and I'm here to give you a snapshot of ValidaCell and one of our core technologies called chemstress fingerprinting and how Lonza are interested in applying this technology. I think 2020 has clearly reinforced the powerful role that science can play in our daily lives. The response of our partners in biopharma to the COVID-19 pandemic has really demonstrated what's possible, but it's also challenged us to keep moving forward with that same vigor. I think today, it's not enough to develop a novel treatment like a biologic or a cell therapy. You have to be able to manufacture it in a controlled and consistent manner in order to get it to that patient. And there are significant challenges that still exist in production of these therapeutics, along with a hefty price tag. At ValidaCell, we are focused on enabling the sustainable manufacture of these medicines by developing analytical tools and technologies to support this process. We've adopted a data-driven strategy to improve process control and make biomanufacturing both faster and cheaper. In order to achieve this goal, we focus on various elements of the process, including accurate characterization and quantification of the protein or cell-based therapeutic, the cell that is the production system for the protein therapeutic, or the media environment in which these cells are grown. Being able to define and consistently measure relevant features within these core elements is central to developing a controlled process. Today, I'm going to focus specifically on the role that cell culture media plays in biomanufacturing, so something a little bit different. Media is one of the most important and one of the most difficult components in biomanufacturing to design and control. It's well established that media used in biomanufacturing should adhere to a defined recipe and manufacturing system. However, unfortunately, in practice, this is not always accomplished. Lot-to-lot media variation persists for a number of reasons, whether that be variation in raw materials, deviation from formulations, or transport and storage-induced degradation. These changes in the media don't only affect the production cell, but they affect the product quality and yield. And as a result, media variation remains a major cause of bioprocess failure. Currently, standard quality control systems for both raw materials and formulated media are based on analyzing the chemical composition of the media that can contain up to 120 different compounds. Another element of this is that cell assays are run where cells are grown in the presence of these media for a number of generations. But we feel that this is only scratching the surface. At ValidaCell, we ask ourselves, where is the biology? Understanding how variation in media affects production is a biological question that requires deep cellular functional profiling to identify how variation affects the biology of the production cell and leads to changes in the product yield and quality. Simple, rapid, and biologically informative assays are needed in order to capture this missing biological data and reduce bioproduction risk. At ValidaCell, we have designed this chemstress fingerprinting technology to tackle this problem. Chemstress allows you to profile a cell's phenotype in a very specific media environment. This is a 96-well plate-based assay that contains lyophilized chemical stressors that have been selected specifically to simulate stresses that a cell might encounter in a bioreactor system. The cells are grown in the presence of the chemicals, and specific biological metrics of interest are measured. This includes maybe product titer and cell viability. And these are used to form a chemstress fingerprint that is specific to that cell in that media environment. A consistent feature of all Valida technologies is that this assay comes in a simple, rapid, and easily automated, cost-effective format. It doesn't require any specific instruments to run this. All you need is a microtiter plate reader, which is a very common and standard laboratory instrument. And we have managed to successfully demonstrate the ability of this technology to reveal similarities or difference in cell culture media environments. You're able to functionally profile your cell in distinct media lots in comparison to, say, a reference lot, and determine whether this has changed. We've also identified a number of alternative applications for this technology, including media development, clone profiling, and clone stability testing. Our work with Lonza through the program has been to investigate the potential use of ChemStress as a tool to direct a media development program. In the same way that ChemStress is able to pick up differences in media lot variation, it's also able to identify bioactive compounds for media formulation by looking specifically at the biology of the cell. And in addition, this year we are working on developing this technology for the cell therapy industry to target initially human mesenchymal stem cell characterization, as well as media development and optimization. I think this is a considerably bigger challenge in the cell therapy space, considering the reliance on human and animal-derived supplements for the expansion of these products. Our overall goal with all of this work is to provide technologies that will reduce time, cost, and risk associated with biomanufacturing by enabling data-driven decisions for improved process control. So with that said, I'd like to say thank you to everybody who was involved and open up for questions.
Perfect. Thank you so much, Stephanie. Always great to see you pitching. We've one of the first questions coming in here, and that is all around, well, the comparable advantage of your solution. So especially talking about the costs of manufacturing. Did you conduct any studies to outline how much that is increased or like decreased and the efficiency is increased using a platform against peer technologies?
Well, as I described, Fabian, a number of the peer technologies don't actually assess the biology of the cells. So one of the experiences that we've had with a number of customers is that they will have grown cells in different media lots and everything looks fine until they scale up to production, and then all of a sudden the cells don't perform as expected. And the cost of that is astronomical. We can't put a number on it because nobody releases those numbers, but essentially what we're trying to do is prevent a bioprocess failure due to media variation or an uncontrolled media environment.
Okay, fantastic. Yeah, I think for some of the competitors, it would not be that feasible to actually like publish those data. One question, and that is more around like the development and the vision for Valida cell. So where do you want to go next? How do you want to develop your platform further?
So at Valida Cell, ChemStress is just one of our many platforms, and we really believe our holy grail is near patient advanced manufacturing that is bespoke to the indication. So from a personal experience, I work in the cell therapy industry, and we have a long way to go in order to manufacture cells in an appropriate way to treat the amount of people we need to treat. So our company's goal really is to drive and power that innovation, to be able to control those manufacturing processes in the way that is required to meet the need currently.
Okay, perfect. Thank you so much, Stephanie. There are a lot of more questions, but I hope they will be addressed during the startup booths and the networking break. Yes. So looking forward to that. So next up from our side, we're short before our networking break. In the networking break, please stay tuned. We're